Hello, everyone, and welcome to today's broadcast, Sample to Insight, Everything You Need to Know About RNA Sequencing. It is presented by Jonathan Shaffer, Ph.D., Associate Director of Next Generation Sequencing Assay Technologies at QIAGEN. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, visit www.qiagen.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Shaffer. I will now turn the presentation over to him.
Thanks, Judy. I appreciate the opportunity to talk with everybody today. And again, we'll be discussing everything you need to know about RNA sequencing. Let's begin with our standard legal disclaimer that QIAGEN products shown here are intended for molecular biology applications, and these products are not intended for the diagnosis, prevention, or treatment of a disease. So we have a packed agenda today. We'll begin with an RNA Focused NGS overview, and then we'll discuss RNA extraction, library preparation, sequencing, analysis, and finish up with a brief discussion about Genomic Services, a summary of today's talk, and then we'll go into any questions that you have. So first off, let's take a look at RNA-focused NGS. So NGS enables a thorough investigation of both coding and non-coding RNAs. This is specifically applied to gene expression, pathway analysis, biomarker discovery, allele-specific expression, fusion genes in cancer, tumor heterogeneity, immune repertoire, as well as single-cell sequencing. Now, of course, there's other applications beyond this, but at the current moment, these are more of the hotter areas of which NGS is involved with. So if you look at the variety of specific analytes, you have the small guys, you have the microRNAs and the piRNAs, so your 20 to 30 base pair RNAs. You have your tRNAs that are slightly larger, all the way up then to looking at your long non-coding RNAs and ultimately your messenger RNAs. And whether you're looking at your microRNAs, which are the master regulators of gene expression, or your mRNAs, you are able to apply any of these molecules to these specific applications. Very importantly, all RNAs are unique and have individual properties that matter. So your mRNAs are coding. There are approximately 200 to 100,000 nucleotides. They have a 5' cap. They're polyadenylated. There's more than 21,000 human genes. And they have the entire expression range, so very low to very high. Long non-coding RNAs are different from mRNAs in that they may or may not have the polyadenylation site. And they also have very low expression to moderate expression. Now, microRNAs totally blow the whole thing up. So they're non-coding, they have a regulatory function, but they're only 19 to 25 nucleotides. As a result of that, there are specialized library preps that are made particularly for microRNAs. And very much like their larger friends, the mRNAs and long non-coding RNAs, they have very low to high expression levels. And an overall tip is that the unique properties of RNAs demand specialized library prep solutions. So when I was putting together this presentation, I came up with this pyramid. So I termed it the Pyramid of NGS Success. So it starts with the bedrock of your sample source. So if you have good sample material or know what type of sample material you're working with, say your FFPE samples, this lays the foundation for the rest of your work. And throughout today's talk, we'll work our way up the pyramid, starting with the sample source, ultimately finishing with analysis. So first off, looking at sample types. Typically, when performing sequencing, your sample types are your fresh or your frozen cells and tissues, your FFPE tissues, serum and plasma, biofluids in general, so cerebrospinal fluid, bronchial lavage, urine, etc., and ultimately even whole blood. And our general overall tip is that we recommend choosing sample prep kits that provide you with the flexibility to analyze all RNAs. So you may say, well, at this point in time, why do you say that? All library prep kits should be created equal, as well as all these sample prep kits as well. And the answer is that some kits still only give you the option of looking at your longer RNAs. And this isn't the best idea, particularly when you're interested also in microRNA. So my advice is always make sure that you pick a prep kit that allows you to look at both your small RNAs and your long RNAs. So this moves nicely now into our second point of RNA extraction. So here at QIAGEN, we've developed our reputation as the gold standard for sample prep over years and years and years. So for fresh and frozen cells and tissues, we recommend the microRNA kits. So the microRNA kits give you the true total RNA fraction, so everything from your very small microRNAs to your mRNAs. These come in three flavors: the mini, the micro, and the 96. Minis for your standard samples, micros for your ultra small samples, and 96 is when you need more than just your single spin columns. You have an entire plate of samples that you can do with the microRNA 96 kit. We also recommend the RNA Plus Mini, Plus Micro, and the Plus 96. As you guessed it, it has the same format, so the minis, the micro, and the 96. And then ultimately, if you're interested in DNA, mRNA, and microRNA, we recommend the All Prep Kit. Now for FFPE tissues, serum and plasma, and exosomes, sort of has the same flavor here. So you have the microRNA FFPE kit and the All Prep Kit for FFPE. For serum and plasma, we have the Chiazol chloroform free solution, the microRNA Serum Plasma Advanced Kit. We also have our long-running microRNA Serum Plasma Kit, as well as the QIAAMP Circulating Cell-Free DNA RNA Kit. This kit is very nice in that you have both DNA and RNA in the same eluate, allowing you to do those multimodal type of experiments. For exosome enrichment and isolation, we have the ExoRNA kit, both the MIDI kit and the Maxi kit, and the choice depends on your input amount. So the MIDI kit gives you up to about 1 ml of fluid, while the Maxi kit accommodates more than 4 mLs. Stuck over here in the middle is the PAXgene kits. So you have the PAXgene blood kit that allows you to isolate RNA from whole blood, and the tissue kit that allows you to, of course, isolate your total RNA from tissue. Now, a point that's constantly brought up over the years is about heparin. So heparin comes into play with your plasma samples. And sadly, heparin is a very potent reverse transcription inhibitor. And for a long time, because of this, you have the inability to use heparinized samples. Basically, you get the cleanest library that you could ever imagine, basically because there's no library there. And in response to this, we have developed a solution to remove heparin from your RNA samples. And if you're interested in this, you can ask a question about this, or you have my email address through the slides, and feel free to email me and I will send you the protocol. Also, when you're looking at sample prep, you often need a variety of instrumentation. So we have the Qpx. So this enables you to process 96 well-format samples. And in conjunction with this is the RNA 96 Qpx kit for isolation. Now your QIACube, this is a 12 sample per run isolation solution, and basically enables automation of any of the QIAGEN spin column kits. I like to say that this is basically an extra pair of hands that you have in your lab. You load your samples on, and ultimately everything is performed that you get your isolated nucleic acids. For quantification and quality control of your DNA and RNA, there is the QIAGEN. The sample input range is from 1.5 to 2,000 nanograms per microliter of double-stranded DNA. Very nicely, there are preloaded apps for the specific isolation kits from QIAGEN. If you're using isolation kits that are not QIAGEN, of course you can still use the QIAGEN. Very nicely, too, you can assess 16 samples in only 2 minutes. Now, looking at the quality control of the RNA sample, so what actually matters? So when you're looking at concentration, I recommend a standard spec for working with either fresh or frozen cells and tissues, as well as FFPE samples. And for serum and plasma, I don't recommend any QC. Now, probably you're like, what in the world is he talking about? So we'll touch on serum and plasma here once we go through both the purity and the size distribution. So for purity, so this is your classic 260-280, 260-230 ratios. Again, we recommend a standard spec. And for size distribution analysis, we recommend the QIAGEN or the bioanalyzer, the tape station, or the fragment analyzer, or something along those lines. And again, we recommend these for cells, tissues, and FFPE samples. And looking very quickly at the QIAGEN Advanced, you can assess the size distribution of up to 96 samples per run, and the speed is about 12 samples for every 3 minutes. And what you receive from this is an RNA integrity score, or a RIS number. All right, so back to serum and plasma. So why don't we recommend taking the concentration, assessing the purity, or assessing the size distribution? And the reason is that in serum and plasma, they are largely devoid of ribosomal RNA. And typically speaking, ribosomal RNA constitutes more than 80% of the RNA in your samples. And as a result of that, when you don't have ribosomal RNA present, your concentration readings look very low. But in fact, they're very much on par with what you would expect with cells and tissues if you remove the ribosomal RNA. Same thing when the 260, 280, 260, 230 ratios are derived. These are derived largely looking at the ribosomal RNA. So if you don't have them there, your purity assessment isn't that valid. Ultimately, when you look at size distribution, typically you're looking at the integrity of the ribosomal RNA bands. And again, without them there, really your risk number is very low, but in fact your sample quality is probably perfectly fine. Okay, so now let's shift gears here again and discuss library preparation. So first off, we'll start to take a look at a whole transcriptome assessment. So typically, whole transcriptome is done using stranded RNA library preps. What does this mean? So say you have the following example. You have a chromosome plus strand and a chromosome minus strand. Gene A is on the plus strand, gene B is on the minus strand, and these two overlap. So you have transcript A and transcript B with this overlapping region. Now, if you don't perform stranded RNA-seq, you don't have any retention of the transcript orientation. So as a result, you can't actually see if this is transcript A or transcript B. Stranded RNA-seq gives you this benefit that the transcript orientation is retained, and you can say if it's actually A, B, or both. And just as a tip, when you're working with whole transcriptome analysis, our recommendation is to start with more than 100 nanograms of total RNA to reduce the potential for duplicates. So for whole transcriptome sequencing, enrichment or depletion is often necessary to maximize your reads from the RNAs of interest. And this is because the typical RNA composition in a cell is more than 80% ribosomal RNA. So if you're interested in ribosomal RNA, this is the perfect solution if you don't enrich it. But of course, most people aren't interested in specifically ribosomal RNA. They're interested in your mRNAs or your long non-coding RNAs. And as a result, you want to perform either enrichment for your mRNA or depletion of your ribosomal RNA to obtain your reads of interest. So what are two styles of strategies? So for Polya Enrichment, basically you're pulling out your mRNAs. You're making use of the fact that mRNAs are polyadenylated, and you basically capture these polyadenylated RNAs. Now, the one caveat of this is that long non-coding RNAs typically do not have a poly-A tail. So as a result, if you're interested in long non-coding RNAs as well, you want to perform ribosomal depletion. And basically what that means is that you are removing the ribosomal RNAs and you are left with your mRNAs and your long non-coding RNAs. And the strategy ultimately depends on what you want. So if you want mRNAs, it's fine to just enrich for those. But if you're interested in mRNAs and long non-coding RNAs, make sure to perform ribosomal depletion. So at QIAGEN, we have two kits here. We have the QIAseq Stranded mRNA Select Kit. What this kit enables is mRNA enrichment and subsequent library prep. And we also have a QIAseq Stranded Total RNA Library Prep that can accommodate samples that have been ribodepleted. Now, if you're looking at the current menu of offerings from QIAGEN, we do not have a ribosomal depletion kit listed on the website. And the reason is because we actually have a brand new kit launching basically as we speak here. It's called QIAseq Fast Select, and it's basically a one-step ribosomal depletion method that can be incorporated into either the QIAseq Stranded Library Prep Kit or any of your common library prep kits from Illumina, NEB, Kappa, etc. So if you want more information on the QIAseq Fast Select Kit, certainly send me an email or ask a question about it. So a little more theory here on how stranded libraries are prepared. So traditionally, we have the following method. You have your RNA and you fragment it. You then perform reverse transcription in the presence of actinomycin D, which prevents antisense reverse transcription. From there, you perform second strand synthesis with DUTP. Following end repair, you have adapter ligation. So you're ligating your Y adapters. And then ultimately, you have your library amplification. And this is basically your second strand is tagged with the DUTP, and this is degraded, and the tagged DUTP strand is not amplified during PCR. So what that enables you to do is that you can have strand specificity because you've basically degraded one of your strands during library amplification. So a second way to derive a stranded library is what we do with the QIAseq kit. So you still fragment your RNA up front, and you perform reverse transcription. However, we use a new method where you do not have to use actinomycin D. In the second strand synthesis, we synthesize the second strand with normal DNTPs instead of DUTP. However, we use a phosphorylated random primer. So then, ultimately, when we perform our strand-specific ligation, you have this phosphate on one strand. So what this enables is that you have ligation of your adapter to this 5' end of the fragment, this 3' end of the fragment, and this 3' end of the top strand. But the bottom strand, 5' end, does not contain a phosphate. And as a result, the adapter arm is not ligated to it. What this means is that during then library amplification, you only have exponential amplification of one strand, and this enables you in a different manner to have a stranded library. So looking at a little bit of data of the QIAseq stranded kit, we used the seqC samples. So this is your universal human reference RNA and the human brain reference RNA. And we also did the standard titrations of 3 parts to 1 part universal human to human brain, as well as 1 part universal human to 3 parts human brain. We used the QIAseq stranded kit as well as competitor T. We sequenced this on a HiSeq 2500. We performed analysis on the CLC Biomedical Workbench. And in the subsequent slide, you'll see the PCA analysis to view the overall expression profiles. So for strandedness here, strandedness is just, again, like I was mentioning, you want to know what strand your library prep is coming from. And typically speaking, you want this to be at about 96 or 95% and above. And both the QIAseq kit and competitor T are both in this range. So what it shows you is even though there's 2 different ways to derive strandedness, both basically accomplish their goal of deriving strandedness. So now what's fun about the seqC samples and their titrations are that when you perform a PCA analysis, they group together in their expected order. So first you have on the ends of the PCA, universal human reference RNA and human brain RNA. So when you have 3 parts universal human to 1 part human brain, this is very close to your universal human reference. And when you have 1 part universal human to 3 parts human brain, this is more towards your human brain. And again, it's closer grouped with the UHRR because this is, again, a universal human reference RNA. So it's packed with RNA from all types of tissues. So when you overlay the QIAseq kit and competitor T, basically everything is on top of each other. So whether you're looking at universal human reference, the titrations, or universal human brain, the samples are on top of each other. So the conclusion that can be drawn from this is that QIAseq and competitor T demonstrate very similar strandedness as well as expression profiles. Okay, so now moving beyond the world of whole transcriptome analysis. So beyond whole transcriptome analysis, you can perform targeted RNA-seq or you can perform qPCR validation. But since today we're talking about sequencing, we'll focus on targeted RNA NGS. So why should you do targeted NGS? Basically, it gives you the ability to have more relevant data. This is because you are defining your targeted regions, because in whole transcriptome, you have regions that are basically not of interest to you. So you're able to basically pinpoint what you want. Also, you have an improved limit of detection because more read budget is allocated to each sample. When you're only focusing on what exactly you need, it gives you an increased confidence in calling your low expression genes. Tied with this is that you can multiplex more samples together. So typically speaking, when you're performing a whole transcriptome analysis, if you've Polya enriched, you need about 25 million reads per sample. However, when performing targeted RNA-seq, and say you only are multiplexing 500 targets together, you need much less read budget for your sample. So the final point, why would you perform QIAseq targeted RNA NGS? And basically because all library AMP and sequencing bias is removed through the usage of unique molecular indices. So here we demonstrate the principle of the unique molecular indices. So let's take a look at your original sample. So if you look at 2 genes, gene 1 has 3 molecules and gene 2 has 2 molecules, giving you a ratio of 3 to 2. So then you perform your library prep and ultimately your sequencing. And ignoring the small colored bars for now, let's just take a look at the raw reads. So after you perform your sequencing, you have 12 reads of gene 1 and 6 reads of gene 2. And this is now a ratio of 2 to 1. You basically go, whoa, like what happened here? So originally you started with 3 to 2, but now you're 2 to 1. And the problem is that during library prep, there are amplification biases, and also during sequencing, there can be sequence biases as well. So to work around that, if you incorporate unique molecular indices into your library prep, basically each individual molecule is tagged with its own identifier. And in the QIAGEN kits, we use a fully random 12-base sequence that gives you nearly 17 million different combinations of UMIs. And what this happens then during analysis is that the reads are collapsed based on the molecule counts. So let's take a look at gene 1, and now look at the small colored bars. So while you have 12 reads, you basically have 3 molecules. You have 4 replicates of molecule 1, 3 replicates of molecule 2, and 5 replicates of molecule 3. And these reads then are collapsed into just molecule 1, 2, and 3 based off the UMIs. Then for gene 2, you have 6 reads, but only 2 molecules, 4 copies of molecule 1 and 2 copies of molecule 2. And again, these are collapsed just into 2 molecules. So when you account for the UMIs, you then retain your original 3:2 ratio. So let's put this in practice here. So the assumption is as follows. So if you have 4-fold dilutions of RNA, so 20 nanograms, 5 nanograms, and 1.25 nanograms, and you amplify these the same, you should actually see 4-fold differences in reads. So on the left here, we have when you just map the raw reads. So what you can see is that actually you have, roughly speaking, the same amount of allocated reads per sample. And the reason is that you are loading equimolar amount onto your sequencer. So ultimately, it will just be read all about the same amount. However, when you account for your UMIs, you're now removing any replicate reads. And when you remove the replicate reads, you see this expected 4-fold drop in molecule counts. And collectively, counting UMIs delivers an accurate representation of your starting RNA amount. This is a typical workflow that can be performed with targeted RNA library prep. So you start with your total RNA, and then you perform cDNA synthesis, just a standard RT reaction. And in step 1, you perform a primer extension and UMI assignment. Basically, you have a gene-specific forward primer with a unique molecular index. Following a cleanup, you then perform a first stage PCR. So this is a target-specific amplification with a gene-specific reverse primer. And this also contains a universal sequence. Following a B cleanup, you then perform a second stage PCR, which enables you to perform library amplification as well as sample index assignment. And this is performed using primers that target universal regions added during the 2-stage target-specific amplification reaction. All right, so now let's shift gears here and move to the small world, so looking at the microRNA sequencing. So if you look at some current challenges of microRNA, there was a requirement for inconvenient sample amounts. So traditional small RNA-seq kits require up to 1 microgram of sample input and may not be ideal with lower RNA input amounts. And this is problematic because there's a lot of effort focused on microRNA sequencing from biofluids. And as a result, this requirement basically renders older kits not compatible or optimized. The second problem is poor data quality. So on-target microRNA NGS account for only about 20 to 30% of your total reads. And often this is okay. However, the traditional kits were only mapping 20 to 30% when it should have been about 60 to 70%. Part of the problem was that adapter dimers are the bane of every microRNA researcher's existence. So these eat away at your read budget. They are very small, they're preferentially amplified, and they're preferentially sequenced. As a result, what you had to do was perform gel band excision. And also one point is that biofluids may be contaminated with similar sized RNAs. And overall, there was a lot of tedious nature with microRNA sequencing. So gel-based workflows can take multiple days, and this becomes very problematic with a large number of samples. So what we've developed is the Seek Micro RNA Kit. It's compatible with Illumina sequencers, and as of a couple weeks ago, it's actually compatible with ion torrent sequencers as well. You can perform differential expression and novel microRNA discovery. What distinguishes this kit? Basically, it's a gel-free rapid workflow. You heard it there, it's gel-free. And so what I like to say, if you're interested in sort of the, the retro style science and you want to be like somebody's in the 1980s performing scientific research, certainly stick with your gel-based approach. But if you want the most rapid, clean way to make a microRNA library, try Seek Micro RNA. This can be done whether you have 1 nanogram or 500 nanograms of input, and with whatever type of sample you have, whether it's serum, plasma, biofluid, cells, or tissues. Interestingly, we've also minimized the presence of HY4 yRNA. And you may say, well, why would you do that? And the answer is that in serum and plasma, HY4 is overrepresented up to 70% or more. And basically, it's the same size as a microRNA. So without eliminating it, it basically eats up all of your read budget. Now, a tip I have here is that microRNA sequencing actually works for any small RNA that has a 5' phosphate and is approximately 50 base pairs or less. So if you're interested in tRNA fragments or piRNAs or other small RNAs in that general size frame, microRNA sequencing will work for you. Here's the workflow. You ligate adapters to the 3' end and the 5' end of the microRNA. And we add unique molecular indices during reverse transcription. And this RT primer basically targets the 3' adapter. At this stage, too, is where the Seek kit eliminates adapter dimers from the library. Following a cDNA cleanup, we then perform library amplification. So here I have represented where we index with a single index. And at the time of this putting this slide together, which was, again, only a couple weeks ago, we had 48 indices. Now we actually just released a 96 index kit. So if you're on the line and listening and you want more than 48 indices, let me know and we can have a conversation about it. And just as a note, this is the style of BA that you will routinely see with the Seek Micro RNA Kit. This is one single nice sharp peak for your microRNA-sized library. Okay, so looking at one piece of data compared to a competitor kit, what I have here is a page gel after a standard library prep protocol. So this is the Seek Micro RNA Kit, a single sharp band. And what this allows you to do is go straight to the sequencer. Now for competitor 1 and 2, you do have that nice microRNA band. However, it's very faint, and it's about the same intensity as an adapter dimer. Also, there's larger weight and lower weight contaminants. As a result, you would need to perform a gel excision prior to sequencing. So for this experiment, we did perform that gel excision on competitor 1 and sequenced the against the Seek kit. So what you see is about 375 reads with competitor 1, but with the Seek kit, you actually see more than 600 microRNAs with greater than 5 mapped reads. So you go, okay, well, that's pretty nice. That's 200 more microRNAs. The even cooler thing is that if you look at the experimental setup, we used only 100 nanograms for Seek Micro RNA and 1 microgram for competitor 1. So we actually used 10-fold less input and still got 200 more microRNAs. Okay, so another shifting of gears here. So what you'll see is that there's a lot to consider when you're working with RNA NGS. And this is something that, I would recommend downloading the recording and listening to this a couple of times. And certainly, we're always here as a resource for you. So we have a new kit called Seek Qpx, and that stands for ultraplexing your NGS gene expression. Basically, we have this for a 3' transcriptome as well as a 3' targeted RNA. It works for samples whether you have 1 to 100 cells, and it's basically a direct lysis method. And it also works for very low RNA inputs, so 10 picograms to 1 nanogram. And basically, we are tagging each of these samples during cDNA synthesis. So to show this a little bit more, how we do this is that we use plates of anchored oligo-DT primers that have the following structure. You have the anchor, you have the oligo-DT, and then you have the UMI sequence. So again, this is a 12-base fully random UMI, and every well has its own unique cell ID. So a 96-well plate has 96 unique IDs, and the 384-well plate has 384 unique cell IDs. And what's really cool about this workflow is that after reverse transcription, you can combine an entire plate worth of cDNAs into one tube. So if you have even up to 96 or 384 samples, you actually only have to perform one ultimate library prep. But what's even cooler is that if you want to have like hundreds of samples or dozens of plates, you can actually tag each of those. And when you incorporate your traditional library ID, you can do, say, 48 sample IDs times the 96 cell ID, or 48 sample IDs times the 384 ID, and do thousands of libraries. Alternatively, if you want this just because you do a small number of libraries but want to still multiplex it, you only even have to do 8 transcriptomes at a time. So basically, whatever type of throughput you have, this solution can work for you. Showing this a little bit differently. So basically, take this 96-well plate and look closely at it with a magnifier, and this is an individual well from the plate. So you have your cDNA, you have your poly-T, you have your UMI, and you have your cell ID. And basically, every well, again, has its own unique cell ID. And what this enables you is after RT, you put everything in a single tube and perform your remaining reactions and library prep. All right, so now that we have library prep done, let's move into sequencing. So first off, there's often the discussion about what read lengths are recommended. So we'll give you some recommendations here for stranded, targeted, and microRNA. But certainly, if you have questions of what else you should be doing, particularly, say, with UPX, you can email me. But very nicely, too, in all of the QIAGEN kits, we put the recommended read structures in the handbooks. So for stranded RNA-seq, the basic protocol is a paired end 2x75, and you can perform a 2x150 if you're looking for novel transcripts, splice site variants, splice site isoforms, etc. For targeted RNA NGS, a single 1x150 is required. And just as a note, other kits may be different. So for Seek Micro RNA, we recommend a 1x75, and this enables you to sequence through the UMI. Other kits recommend a 1x50, but they're a little bit behind in that they don't contain UMIs. And if you're used to only using a 1x50, and if you don't care about using the UMIs with the Seek kit, you can still just use a 1x50 with the Seek Micro RNA Kit. And as a tip, always check whether kits require custom sequencing read structures and or custom indexing primers. Second point is read allocation. So for stranded RNA NGS, we touched on this earlier, but we'll talk about it again. When you select only for your mRNAs, you need about 20 to 25 million per sample. Ribosomal depletion, you need about 50 million reads. So looking at targeted RNA NGS, this is the value then of a better usage of your read budget. For shallow reads, you need about 2,000 reads per gene, moderate 5,000 reads, and high coverage only about 15,000 reads. So again here, if you have a small panel, you can make very efficient usage of your read budget. And for microRNA NGS, a general catch-all is about 5 to 8 million per sample. For your FFPE and difficult fluids, we recommend 10 to 20 million, and for fresh and frozen, less than 5 million. So a couple tips here. So kits with UMIs can help you optimize your read allocation by revealing the amount of read replication. Okay, so if you're performing RNA NGS and you're reading every molecule 50 times, that means you're basically over sequencing your sample. And then in subsequent experiments, you can back off of your read allocation per sample and make more efficient use of it. Tip 2 is I always like to oversequence, particularly when you're working with tough samples like FFPE or difficult fluids. Cerebrospinal fluid or cell culture media can actually be quite difficult to sample. So it's always best to allocate as much reads as you can. Tip 3, the number of samples that you can multiplex depends on your application, your sequencer, and your reagent cartridge. Now, I know sometimes this goes without saying, but importantly, make sure that you know all the moving parts when you're considering what number of samples to sequence together. For example, MiSeq is a great sequencer, but you only have about 30 million reads. So if you have 10 FFPE samples, probably it's best to use a sequencer with a higher capacity, such as a NextSeq. And another recommendation involves the amount of library to load on your sequencer. And here we'll just look at Illumina instruments. And as a note up front, ultimately the optimal amount may need to be empirically determined. However, some basic guidelines are that for the QIAGEN stranded, 9 picomole on the picomolar on a MiSeq, or 1.6 picomolar on a NextSeq. And for both QIAGEN targeted RNA NGS and microRNA NGS, we recommend 10 picomolar or 1.2 picomolar. And again, all sequencers are slightly different. Even if you have optimized your amount of loading and you then move to a second sequencer, all sequencers are slightly different. And be sure to find your sweet spot for the loading and err on the side of caution. So now let's move into analysis. And I'll preface the analysis with, I'm a bench scientist. So typically speaking, my expertise is in RNA extraction, library prep, and sequencing. And why I say that up front is that the analysis can often be a tricky thing. So what I always typically look for is prepackaged analysis solutions that basically enable you to move your lack of expertise aside and allow the bioinformatic experts to analyze your data. And a lot of times you can actually do this on your own using pre-designed pipelines. So for stranded RNA NGS, we recommend the CLC Biomedical Genomics Workbench. And basically, it allows you to analyze and visualize data from all major NGS platforms. And here we have a workflow picture of CLC at the bottom. There's basically ready-to-use workflows for all types of analysis, whether you're looking for whole genome sequencing, whole exome sequencing, targeted sequencing, whole transcriptome sequencing, even microRNA sequencing. There are pre-designed workflows. And for people like me that don't have the bioinformatic expertise to design your own pipeline, these are very, very favorable. In addition to CLC for targeted and microRNA NGS, we also have a couple free analysis packages through the GeneGlobe Data Analysis Center. And looking at this as one example for microRNA, basically you upload the data, and then the data analysis is performed for you. And this is available at QIAGEN.com/GeneGlobe. And what this site does is it performs adapter trimming, read mapping, and UMI counting. And it's very much as easy as uploading your files and clicking Start. And what you receive is a variety of outputs of your data. You have a summary tab that gives you important mapping metrics for the sequencing run. You have a microRNA piRNA tab, which basically gives you the entire list of microRNAs that were found, as well as their UMI counts, as well as their read counts. In addition, we do this for other RNAs and not characterized mappable. Not characterized mappable is basically a tab that you can use if you want to look for novel potential microRNAs. And again, what we're giving you is a list of the sequence, list of the genomic coordinates, as well as the UMI and the read counts. And from there, you can take this information and basically start the assessment of whether it is a real microRNA or not. Following primary analysis, we also have secondary analysis. And secondary analysis is your differential expression analysis. There's a variety of ways to normalize your data, including GNorm, total molecular tag counts, DSeq2, and trimmed mean of M, and basically you get a fold regulation output. For tertiary analysis, we have the ingenuity pathway analysis. Basically, it's asking what's next by modeling, analyzing, and understanding complex omics data sets. And what I like to think of it is that it is a very much automated PubMed system. Break it down basically into its roots. Okay, so what IPA does is that it collates and collects and quality assures all basically publications and data sets that are deposited into the public database, and it integrates them together and provides you connections to all of your papers. Okay, so instead of manually looking paper by paper by paper by paper, it basically, if you upload your data set, it integrates this into the IPA set and basically weaves it into what already exists and what is known for the data sets. We also have the OmicSoft Array Studio, and basically you can go from FastQ to Insight. And data processing is performed in the OmicSoft Array Studio, and you can move from FastQ to your differential gene expression analysis. In addition, if you look at IPA and analyses of OmicSoft lands datasets, and we'll go over that in a minute, on the OmicSoft side, you have processing, curation, and QA of hundreds of thousands of RNA-seq samples integrated into the OmicSoft lands. And these would be your Oncoland and your disease land with more than 8,000 comparison data sets. As I already mentioned, you have your journal articles and any deposited data sets into public data sets, and you have curated findings integrated into IPA. So when you combine OmicSoft with IPA, you can have the biological analysis of each of your data set and comparison of your analysis to all of the OmicSoft analysis. So basically, you take your data and you do the tertiary analysis. It gives you what's already known, what's potentially unknown, and literally gives you a built-in future direction for your experiment. And a quick look at array lands. This is basically where you can store, manage, search, and visualize basically any of your data. You can analyze millions of genomic samples across multiple platforms, and there is also hosted data content, including Oncoland, Disease Land, and Genetics Land, but you can also have your own privately customer-built lands. All right, so let's finish up here. We'll take a quick look at Genomic Services. And first question is always, why Genomic Services? Basically, Genomic Services does all the difficult parts for you. So if you don't have the time or you don't have the resources to do your experiments, have the experts do them. So we have unique access to R&D and manufacturing, and there's labs in either Hilden, Germany, or Frederick, Maryland. Importantly, there's also access to the innovative QIAGEN technologies, and never underestimate that there's always consultation and guidance available from lab scientists that are actually conducting your experiments. In addition, you have access to people like me that have built the NGS kits, and also my colleagues that have built the qPCR kits, built the sample prep kits, built the automation, etc. So what types of services does the genomics group provide? They provide sample preparation services, library preparation services, Sanger-seq services, qPCR services, custom services. And in addition, they also perform data analysis and interpretation. So here we have Explore RNA, which is a custom-built pipeline for the genomic core analyses. They also have access to Biomedical Workbench, CLC Genomics Workbench, IVA, as well as IPA. So we went over IPA, and basically what the genomics core can do is your entire sample prep, your library prep, your primary analysis, your secondary differential expression analysis, as well as ultimately your tertiary analysis. So looking at, again, what is provided for RNA specifically, there's RNA isolation from a variety of samples, mRNA sequencing and microRNA-seq, whole transcriptome, as well as targeted RNA analysis. And for reporting and data analysis, we already touched on this, but you receive a comprehensive final report that's easy to read and it summarizes all of your results. And as well, you have a follow-up call that is built in to discuss your analysis as well as any next steps. And sometimes I might even be on that call. Just depends on sort of what type the next steps are, so we can give you additional recommendations. So in summary here, we'll go back to our pyramid of NGS success. So if you look at what NGS success depends on your sample source. So the quality matters. So when you're looking for your samples, you want to be sure that your sample material is tightly controlled and homogeneous across your entire study. This is particularly important when working with biofluids. So you want to be sure that, say, perhaps your collection technique is consistent across your entire portfolio of samples. One step up is your sample. So you want to be sure that your type of sample is tailored to your experiment, so cells, tissues, FFPE, and biofluids. Biofluids is an interesting thing. So a lot of times, one of the most easily accessible samples is a whole blood sample. However, whole blood has quite a high background of signal present simply because you have blood cells. So if you're looking for a circulating signature, you want to often set aside your whole blood sample and process it into a serum or a plasma sample and look basically for your cell-free RNAs. Extraction kit is the third level. You want to be sure that you choose a kit that gives you flexibility. Flexibility in terms of both meeting the needs of your experiment, but also keeping in mind what's next for your experiment. So say you have this awesome whole transcriptome analysis finding and your IPA analysis suggests that particular microRNAs are regulated with it, and you did just a large RNA prep. Now you have to go back and re-extract to look at your microRNA. So again here, choose an extraction kit that gives you the entire gamut of RNA from your small RNA to your large RNA. Library kit. Library kit depends on your intended application. And regardless, when you're performing any bead cleanups or size selections, etc., be sure to remove all traces of ethanol. Ethanol is a killer of downstream enzymatic reactions. And in addition to killing the downstream reaction, it also increases your adapter dimers. So, yay, not only have you killed your real product, you've also increased your background product. So just be sure that everything is ethanol-free. Sequencing, pay attention to your details, pay attention to your read allocation, pay attention to your read structure, pay attention to your amount of library that is loaded to your sequencer. And finally, analysis is of paramount importance. You can have the, you can have the best quality samples, the best isolation kit, the best libraries, and if the analysis is bad, everything is stuck and you can't reach ultimate NGS success. So be sure to perform proper primary, secondary analyses, and also keep in mind your tertiary analyses that enables you to add the layer of biological interpretation. Okay, so with that, I think we'll take any questions. I certainly appreciate you taking the time today to listen to us, and feel free to ask any questions. And also feel free to write down my email, jonathan.schaffer@QIAGEN.com. I freely give out my email simply because here at QIAGEN, one of our goals is to give you what you need for your experiments. So if you need help in setting up your experiments or help in determining what isolation kit or what library kit you need, send me an email. In addition, we also have expert technical scientists at QIAGEN, and you can contact technical support at QIAGEN as well. So with that, we'll finish up. Thanks a lot.
Thank you, Dr. Shaffer, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, when performing transcriptome sequencing, do you recommend mRNA enrichment or ribosomal depletion?
That's a great question, Judy. So it comes down to what you're interested in. If you're interested in mRNAs only, I would recommend mRNA enrichment. So basically select for your RNAs that have a polyadenylated site. But that being said, if you're interested in long non-coding RNAs as well, I would recommend ribosomal depletion. The final point is that for heavily fragmented samples like FFPE samples, sometimes the poly A tail is lost. So as a result, it's better to perform ribosomal depletion instead of relying on potentially missing a target because the tail is no longer there.
Thank you. Does QIAGEN offer a ribosomal depletion kit?
That's a good question. So we actually are just launching a kit right now. It's called QIAseq Fast Select. It's a single-step ribosomal depletion kit. You basically add a reagent to your RNA and ramp down temperature for 20 minutes, and it eliminates either your ribosomal RNA or your globin RNA.
Thank you. Does the Seek Micro RNA Kit work with serum or plasma?
It works with both. And this is an area, this is a great question to ask because a lot of effort with microRNAs with serum or plasma now. I often get asked also whether serum or plasma should be used, and basically whatever you have access to as long as it's all serum or all plasma. So I wouldn't recommend working and comparing serum to plasma just because they are inherently prepped differently.
And how much starting material do you recommend?
Typically speaking, we start with 200 microliters of serum or plasma. Comfortably, you can go down to 100 microliters. Mildly uncomfortably, you can go down to even 25 microliters. But all things being equal, I would start with 200 microliters. And if you're working with exosomes, I would probably start with 1 milliliter, just because the amount of RNA should be even much lower because you're looking at a subcompartment.
Thank you. So we have time for one more question. Do you have to do a full plate of samples for QIAGEN Qpx, or can you use a portion of the plate?
That's a good question as well. So in the time we've released this kit, this question has came up repeatedly, so I'm glad this was asked. And the QIAGEN Qpx kit, you have to use only minimally 8 wells of the plate. And what this allows you to do is that you can basically just use a portion of the plate, and the plate is actually a breakable plate. So you can literally crack off a column of wells and just perform 8 transcriptome analyses at one time, or you can perform 24, you can perform 96. So it gives you quite a bit of flexibility to efficiently work through your samples.
Very good. I would like to once again thank Dr. Shaffer for his presentation.
Thanks, Judy. I really appreciate it.
Oh, you're welcome. I would also like to thank QIAGEN for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 11th, 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.